Hello everyone. I am Neha Avasthi of MSc Biotechnology here to present research review topic genetic and epigenetic basis of SCID through multi-omics approaches. Analysis of the molecular etiology of SCID has led to the important insights into the control of immune cell development. SCID is the most severe form of primary immunodeficiency disease. It is characterized by abnormality in the function of immune system due to the genetic defects in the development of T cells. SCID is seen in infants and is fatal if left untreated for the first year of life. Here is a data from the primary immune deficiency treatment consortium, which is 47 centers around the US, you showing that treatment is very successful if given at the younger age. Less than 35% before baby months before babies have started to get serious infections. But these infections begin and they certainly compromise survival, and this is even modern data in the latest decades showing survival decreasing to around 50% if infants have an active infection at the time of their bone marrow transplant. Fortunately, now we have newborn screening for SCID, and it's based on detecting these little circles of DNA called TRECs that are produced from the recombination of T cell receptors that has to cut and paste different pieces together in order to generate the T cell diversity that helps us fight infections, and this byproduct of T cell receptors rearrangement produces a very handy analysis for newborn screening. And this graph shows that newborn screening has increased in the diagnostic methods of SCID. Another determinant for survival of SCID is the genotype. There are many different genes that interrupts the graft. Again, interpreted in graft again from the data of primary immune deficiency treatment consortium. It that is that the survival is the worst for the gene called DCLRE1C. It's called the ARTEMIS gene, and mutation in this gene that produces SCID that has the worst survival of any of the SCID genotypes. The gene encodes a DNA repair enzyme, and this repair enzyme is necessary in order to accomplish the T and B cell receptor rearrangement that are required to make these T cells and B cells mature. So this is called a T minus B minus form of SCID, and because DNA breaks can't be fixed, these individuals also have increased sensitivity to radiations. This type of SCID has the poorest survival because even after the successful donor transplant, B cells immunity is rare, due to which the T cell transplant is incomplete. Secondly, alternative donor transplants have high rates of rejections, poor immune reconstitutions, and increased graft versus host disease. Thirdly, fourthly, the high dose alkylator chemotherapy leads to the short status, poor tooth development, and increased mortality, due to which gene therapy may offer a bit. Another type of SCID in this is the CD45 deficient SCID caused by the uniparental disomy. This type of infants uh, surviving CD45 deficient SCID patients. UPD refers to the inheritance of two copies of chromosome for segment of chromosome from one parent. These are mutations that will be deleterious to protein functions using SVM The number of parentheses quantifies the likelihood that a mutation will disrupt protein functions. The cause of SCID in infants is lack of protein tyrosine phosphatase CD45 that was identified through SNP array and whole genome sequencing. Materials and methods. Serum antigen levels were determined by nephelometry. Lymphocyte proliferation was assessed by measuring thymidine incorporation into mononuclear cells following culture with optimal concentrations of indicated stimuli. Then cell sorting is done by as the mutated and healthy cells are used for RT-PCR and indicated cytochip analysis were isolated from frozen peripheral blood mononuclear cells PBMC using FACS vintage by sorting cells incubated with unlabeled mouse antibody to HLA-DR2 followed by incubation with normal goat serum and staining with RPE goat mouse anti-mouse antigen. Sorting is done because of available patients frozen PBMC obtained post bone marrow transplant, but before T cell development and contained CD45 maternal lymphocytes. Then isolated RNA is used for cDNA synthesis in RT-PCR analysis, and CD45 and beta actin transcripts were then quantified by RT-PCR with the master cycles in apendropsis. Then followed by CD45 sequence analysis is done. In this PCR amplified with primer pairs that generates overlapping products spamming the full length CD45 variant 1 transcript coding sequences. Then total genomic DNA individual amplified DNA samples were each purified, fragmented, biotin labeled and hybridized to a cytogenetic whole genome array. After that, exome sequencing and bioinformatic analysis is done. The metadata are collected and the DNA libraries are prepared. Sample preparation and sequencing were done by expression analysis. Single nucleotide variants and indel variants were identified. A database created from the annotated datasets. For the maternal UPD analysis, alleles that fulfill the requirements were identified, and the requirements were heterozygous in mother's sample, homozygous for wild type alleles in father's sample, then homozygous for wild type alleles in other unrelated samples in the datasets. Then homozygous for alternative alleles in child sample. Another type of parental UPD analysis were done by the requirements or followed by heterozygous in father sample, homozygous for wild type in mother sample, then homozygous for wild type alleles in unrelated samples of datasets, followed by homozygous for alternative alleles in child samples. The tools are used for identifying high confidence mutations from the raw sequence data. For the phenotype of missense mutations, sequence based and structure based informations on each mutations. PSI BLAST is used for structure for homologs for the uniport sequences in the database and to calculate the position specific scoring matrix for each sequence. The homologs with greater than 35% sequence identified to the queries were allied through alignments, and the score for each sequence positions is calculated. Finally, mutations were mapped to protein structures using profile alignments search methods. Moving to results, with patients that lack CD45 expression, when gone through the flow cytometric analysis through peripheral blood lymphocytes at the presentation, revealed normal numbers of B and NK cells, but dramatically reduced number of T cells. Moreover, a few T cells present were found to be non-functional as they were unresponsive and mitogenic stimulation to mitogenic stimulation. CD45 is essential for T cell development and T cell receptor signal transductions and had previously been identified in two fatal cases of SCID. The Sanger sequencing revealed that the patient's mother was heterozygous for a nonsense mutations at the position 1618 of the coding sequence in exon 14 of the CD45 gene that created a stop codon at amino acid 540. However, no mutations were observed in the coding regions of either paternal CD45 alleles. So, this finding suggested that either one copy of the parental chromosome bore a microdeletion elementing the CD45 locus, and the patient inherited two copies of the mutant maternal CD45 alleles. This is the Sanger analysis of the CD45 analysis of the patient and the parents. The schematic depicts the domains of CD45, including alternatively spliced exon that gives rise to RA and RO informs, transmembrane domains and phosphatase domains. The position of nonsense mutations is marked, and the change in nucleotide sequence and amino acid sequence at the bottom in the patient. The Sanger sequence traces on genome DNA from the parent and patients can be seen in. It is caused by the duplication of the mutant alleles. SNP arrays are performed on genomic DNA from EBV lines derived from B lymphocytes of parents and patients, resulting that there are no changes in the copy number across CD45 locus on chromosome 1, due to which the patients had either inherited two entire copies of maternal chromosome 1 or two copies of the region compassing the maternal CD45 mutations. Whole genome array analysis revealed that disomy was also evident in other primary blood cell types, which are derived from embryonic mesoderm as well as in apical epithelium, which is derived from the embryonic ectoderm. In this graph, we can see that copy number and allele peak analysis of various cell populations from the patients are seen. Analysis of copy number and allele peaks in EBV immortalized B cell is depicted. The y-axis depicts the DNA number, copy number, and the allele peak. Allele peak panels normally shows three bands representing all homozygous and heterozygous allele calls. Note that the copy number panels reveal two copies of each gene on chromosome 1, but show only two bands with loss of middle heterozygous and across the entire chromosome, indicative of UPD. Chromosome indicates for each autosome and X chromosome are shown on the x-axis. Copy number and allele peak analysis are depicted for chromosome 1, neutrophil, lymphocytes, and buccal epithelium from the patients. All these analyses are performed using whole genome arrays only. So, due to this, SCID can be treated by gene therapy, and bone marrow, but more specifically gene therapy because through that complete form of B cells and T cells transplantations can take. That's all from my side. Thank you.